Like, everybody stand up for 5 seconds, please, right? And like, just stretch it a little bit. We've been sitting for too long. Okay, good. Just, thank you. Okay, great. Thank you for coming. It's a Saturday. We usually do not have seminars on Saturday in Europe. And so, yeah, I'm really super excited that you all made it, and it's like a big audience. So I'll talk about some things that we have been doing, and my lab mostly is interested in structure of membrane proteins. The reason for this is obviously that we all know that they're very important. There are a lot of drugs that go against membrane proteins, and membrane proteins, while they make a large fraction of proteins in the body, the depositions of membrane proteins in the structural database is only about 4%. So it's not a lot. And the reason for that is that they're notoriously hard to work with. You need to purify them from membranes with detergents, and then they have to go on purification on columns for multiple hours, and they don't like it. Right, so they often aggregate. They are very unpredictable purifications. If you have a good protocol, then the purification may work one time, but another time it may just not work. And I see that a lot of you say yes to that, so I guess you have some experience with that. Obviously, yeah, and then once you have a structure, it can tell you a lot about the protein function, which we're mostly interested in, but also about drug design opportunities that you can have. And so what my group is particularly excited is that there are a lot of undiscovered ground in the membrane protein structure research, is that lipids, transmembrane gradients, and interactin partners actually critically regulate structure and function of membrane proteins. Here is one example. It's the voltage sensor, and depends in which conditions the structure was solved. This transmembrane helix moves up to 8 angstrom. Obviously, 8 angstrom is a huge difference, like in the structure, but also like in the function, because typically what ion channels do, they open the pore and it's a 2 angstrom move, right? So if you move up to 8 angstrom while your functional difference is just a few angstrom, means like you don't really know a lot about your system, right? Okay, so, and what we do essentially, we try to address this question about lipids and transmembrane gradients and native interacting proteins by 2 methods, which is single particle Cryo-EM. Becky showed you. It's all you all very well know this method. But we do try to add membrane mimetics, so which means that we will cover the transmembrane domain of our protein with a special scaffolding protein that would allow adding the lipids to it. I will give you an example on that. And the second thing that we're very excited about is to do tomography on membrane systems, and then together with focusing on ion channels. And this allows us to potentially not purify our proteins and solve structures in native membranes. And I'll show you one example. So essentially, I'll show you 2 examples for this. And our lab is also tech heavy, because tomography and averaging from tomograms is a very developing field. And a very developing field means it's not very well done yet. So we do a lot of work in order to make it easier, first for ourselves, but then we try to release it on our GitHub. Okay, so the first protein I want to show you today out of 2 is the serotonin receptor ion channel 5-HT3. So it's a cation channel that's located in the central and peripheral neuronal system. So it has an ion pore in the membrane, and then once a serotonin molecule, which is a neurotransmitter, binds to the extracellular domain, the ion pore opens, which allows cation flow inside the cell, right? And basically there is a lot of regulation around it. So it was known from physiological electrophysiology, for example, that phospholipids and cholesterol are required for regulation. And for example, a related acetylcholine receptor without cholesterol can bind ligand, but it will not open the pore. And then we have been working on this protein for several years before 2 papers in Nature came out with the structure of the receptor in presence and absence of the ligand serotonin. These were good structures, but what often happens with ion channels is that by looking at the structure and by knowing the conditions, you don't necessarily know what state you have captured. And so you typically need molecular dynamics simulation or some other evidence to actually state what your structure is. And so then molecular dynamics simulation is a little tricky. So it depends on how you do this, you can end up with different evolution of your system. And then this can be debated, which also happened in the case of these structures, that super long molecular dynamic simulations actually question the assignment of those structures to the open state. Also, computers get better over time, and what people could do in 2008, what people couldn't do in 2018, we now can do easily on a more simple cluster. Right. So what my group in Frankfurt with Yinyi and Patricia, who were very, very good and obviously already left the lab, we decided to add another dimension to the system, which was the reconstituting the receptor in lipids, which were the native lipids, brain polylipid that contain cholesterol and phospholipids, but also add membrane mimetics. And we added saposin and nanodiscs. This is essentially, these are polymers that wrap around your transmembrane domain and allow to have several layers of lipids around the transmembrane domain of your protein. And then after this, you basically go through gel filtration, and you can treat your protein pretty much as a soluble protein. So it doesn't have any detergent anymore. And it looks a little bit like this. So you see there is a little nanodisc imitation, like over here, right? Yeah, so an image that looks a little bit like this. I'll also show it. I hope you can see it, that it has little individual proteins. It has a bit of background, but mostly we had a crowded micrograph in order to be able to record data quickly, to record basically more particles per micrograph. Because at the moment we were using Falcon 3, and it's, as Becky showed you, it's kind of slow. But yeah, so from those grids we could record three structures in two conditions, one without serotonin, so two structures without serotonin, and turned out that one structure had full C5 symmetry and another one had the C1 symmetry. So while the structure with serotonin was asymmetric and had slightly higher resolution, 2.8 angstrom, but all the structures had similar resolution. Also, all the structures had density, which could be assigned to lipids. So, for example, this is the density that looked like cholesterol, and it also suggested to so cholesterol was also suggested to bind somewhere there. And yeah, we can see that actually it forms a rather dense network of interactions with the transmembrane helices of the receptor, and this happens in all structures. What doesn't happen in all structures, but we could only see this in presence of serotonin, and is there is a phospholipid that was inserted between the two subunits of the receptor in the transmembrane domain. And when we looked at the same location in the absence of serotonin, we could see that this bright blue helices are just closer to each other. So there is not enough space for the phospholipid to get in there, which suggests that basically the phospholipid gets inside there and stabilizes the open conformation in a certain way. Very interesting, we compared our apostructure to the structures that have been previously solved in detergent by the other groups in those two Nature papers. And so what we could see that central helices that organize the pore, which are M2, they were slightly different in their locations, but the outer helices were significantly different. So the difference was up to 12 angstrom of dislocation. And again, as I mentioned, the functional differences are few angstrom. 12 angstrom is a big difference for an ion channel. I think one of the most exciting things about these structures and comparing them to the structures in detergent was as follows. So ligand binds over here, where the red circle is, and the ion pore is here in the transmembrane domain. So this is the difference between the apostate without the ligand and the ligand bound state. So serotonin is over here, between the apo and the serotonin bound structures, right? So you see that when we add serotonin, the ligand binding domain becomes more compact, right? So you see that side chains rearrange closer to the serotonin molecule, and then this leads to the conformational propagation of conformational excitation all the way down to the transmembrane domain. So here you see the difference between serotonin-bound structures with presence and without, with presence of lipids and without them, and you see that basically in the absence of lipids in the transmembrane domain, the ligand binding domain does not become more compact with when it's added, right? You see that the orange ligand, the side chains in the orange structure are less compact around the serotonin. This means, interestingly, that in the absence of, so basically not only the ligand binding domain regulates the opening of the transmembrane domain, but also the absence of lipids does not allow the ligand to bind properly.
Mikhil.
Yeah?
Yeah. So in the Apple state, you actually saw that, I mean, it's a different conformation than that paper is from Sudha Chakrabarty's lab, right?
Yeah.
2018
Well,
paper.
there are two different ones. One of them is yes.
Right. So do you not think of a possibility where the apostate is actually a different apostate compared to the real closed state? Can it be possible? Because there can be a number of conformations possible, right?
Yeah, yeah.
Like,
So
can you, I mean, like it's always possible to capture a different ground state compared to what has been captured in detergent.
I generally agree with that. The way we think about structures is a little bit static, right? But generally, if you look at any MD of any membrane protein, it's not static. It fluctuates around. So it's very possible that we capture, that one group captures one of them of the local minima, another captures another local minima. That is possible. However, we try to imitate the protocol. So there is a hierarchy of protocols, right? So they use the protocol that goes around the crystal structure that has been crystallized before, right? And then they just used a similar protocol for a CryoEM study. We essentially tried to use a similar protocol and add lipids.
I'm actually having a problem. I mean, specifically, I faced a similar problem. That's why I'm raising this question. So two structures solved on the closed state structures of ACHRs, where it was claimed that these are high resolution closed state structures, but when we did molecular dynamic simulations on them, I'm not able to actually take it to the open state in one of the cases. So that could mean that it's probably locked in a D state rather than a closed state. And your resolution, time resolution of spraying and everything is not actually enough to tell you whether it is actually residing in the closed state or in an intermediate state between closed and D. So that is one problem. And second thing is, now this lipid binding into the M3, M4 that devices, that can, I mean, like, how do you infer this long distance interaction between the pore and the binding site? Because, like, we have seen there is actually in functional electrophysiology, there is no change in energy. Like when you mutate this this lipid binding site, because we have seen actually.
You mean this one, right?
Yeah, that site. So when you mutate those residues, it doesn't change anything at the ligand binding, at the binding site. So there is no functional change in electrophysiology at least. So I'm not sure how you are seeing. So could it be just just a result of this kind of a binding, or it could be a cause of this? That is something I'm not sure.
So I don't remember the literature very well. It's supposed to be the allosteric modulator.
Right.
Right. So obviously it's not directly related, and it's partially optional, I guess.
Yes.
If you can.
So lipids are more flexible and it's allowing the dynamics of the protein, whereas your nanodisk is fixing it in one conformation, it's not allowing much changes. Is it true, correct, what I'm understanding?
The lipids. Well, you would. There is no good like basic science around what happens inside nanodisks. It's very hard to calculate. You would assume that lipids at least could move around a little bit, and so obviously this goes inside when the channel is open.
I think let Misha finish his talk.
I'll have a couple of comments on the methods as well in the end, right? So we did MD as well, right? So what we tried to do, we did this in collaboration with a MD lab, Shuguan Yuan from Shenzhen Institute of Technology. So we tried to do unbiased MD without the transmembrane voltage. And so we did this for the previously reported structures and for our structures without ligand and with serotonin. So we could see that only in our structure with serotonin, the pore was wide enough to pass sodium, and in the other structures we couldn't see that. So we kind of suggested that this is the structure that is more open, but it doesn't seem to be excessively open to be unphysiological, right? And then when we do the toggle between, so it's not MD, it's morph between the open and the closed structure. Like these are the structures in detergent, and this is our structure. So you can see that the one that we have in the lipid disk, I'll play this again, the opening of the channel happened more in the horizontal plane. Doesn't play anymore. It opens in the plane while this structure basically the outer helix moves a little bit up or down as well. And so this is possible that it happens in the native membrane. However, my intuition would be that the protein is more displaced in the plane of the bilayer. While such a displacement that would assume that modification of the thickness of the bilayer is not impossible, but it will assume that there is more energy deposited in the elastic energy of the membrane, right? So, which two structures are correct? I don't know, but like intuitively, our structures seem to be more intuitive because they open in the plane of the membrane. And then again, we always have this question, right? How do you know which one, what you captured, which one is correct? And then this happened recently. Because Cryo-EM is new, and we first started solving those structures, we were very excited about this, and then more labs started to solve the same structures in different states with similar protocols, but slightly different ones. And then, for example, this is an example from Eric Guo's lab. They worked on glycine receptor. And so they tried to go for the open state, and what they've seen is that if they have a nanodisk, which is one type of the membrane mimetic, and they add glycine, then the channel doesn't open, so there is a constriction over here. While if they use the SMA, which is another membrane mimetic, then they would get the open state in presence of glycine. But some of the states, so they would have obviously several states, they would be able to do the classification. Some of the states would have the pore so wide, then it probably would be non-physiological. So this means that nanodisk probably likes to move the protein to the more closed state, while the SMA probably the effect of the SMA is more that the pore is a little bit more easy to open. Like there's more research. This is a recent preprint that shows that the same pentameric ligand-gated ion channel, bacterial one, in presence of different types of the membrane mimetics, actually has a different pore profile. And it's not very different, but it's significantly different. And the pore is really the heart of the ion channel, basically, because this is what defines the way that water and ions would pass through it. So this is without any ligands. It's just the different structures in presence of different membrane scaffold. And obviously it gets a little bit more divergent at the periphery. So obviously then these membrane mimetics, they are good, and as Rebecca said, it's better to have a structure in membrane mimetic than probably without it. And then when we had detergent structure, it's better to have a structure in detergent by Cryo-EM than probably to not have it, or to have a crystal structure that is definitely locked in the minimum energy state. But so I think what we can do, what we can think now is that what is the next frontier in membrane protein research? And I think in some cases, nanodiscs or membrane mimetics do not seem to distort the protein. And it's probably a lot of cases. So in some cases, like in the pentameric ligand-gated ion channels that I've shown you two examples, and also like as I showed you for the serotonin receptor, it seems to play the role. And obviously my intuition is because the protein likes to displace helices in the plane of the membrane, and then you have a belt around it, it just limits you to open. So one way to do the next step to get better structures, or maybe even perfect structures, you can say, that would be to image the proteins in unrestricted lipid bilayers. So if you can do that. And so our lab started to now, once we moved to Berlin a year and a half ago, we started to do this research on trying to image those receptors in unrestricted lipid bilayers. I don't have research on that because our resolution is not very high, but I want to show you something that we got in the meantime. And this is a different protein, so I'll show you a ryanodine receptor, which is a part of the excitation-contraction coupling in skeletal muscle. So the biology is a motor neuron basically initiates an action potential on the surface of the muscle cell, and it goes into an invagination that's called the T-tubule, so this action potential activates voltage-gated calcium channels, which are called DHP receptor, that allow calcium flow inside the cell. This calcium and the physical interaction of DHP receptor with the ryanodine receptor opens the pore of the ryanodine receptor, which is calcium permeable, and it releases calcium which was stored inside the SR vesicle. And then this calcium then leads to the contraction of actin-myosin filaments and then the muscle contraction as a result. So what we're interested to look is this super complex between, which is the interaction between the DHP receptor and the ryanodine receptor. Structures of purified channels have been solved several years ago in detergent, and some of them already in nanodiscs. But so we wanted to really look at this physical coupling between the two of them. And also the interesting thing about the system is that there are 4 DHP receptors per 1 ryanodine receptor. Thank you very much, that's better. But it only happens, basically these DHP receptors are only coupled to every second ryanodine receptor. So there is like a little checkboard. And then obviously, because ryanodine receptors do form lattice on the surface of the SR vesicles, it's also interesting, we would hypothesize that not only calcium and physical interaction with the DHP receptor regulates ryanodine receptors, but also the neighboring ryanodine receptors that literally touch their neighbors also would have an effect on this. So we started this work quite a while ago trying to answer all those questions, and again, we haven't answered any of those yet, but I'll show you how we aim to go about it. So in order to experimentally address those questions, there is an old protocol to purify this T-tubule complex with the sarcoplasmic reticulum, and it has been used before us. And so my student Van Bochen slightly optimized it in Frankfurt. You start with a fresh rabbit muscle, and then you go through several purification procedures which do not purify the protein. You just purify vesicles that are basic, that you can get produced by homogenization of cells. And then one of the bands would have both DHP receptor and the ryanodine receptor, which we confirmed with blotting. And then this you can just take and put on the grids and make a Cryo-EM grids, right? Just by plunge freezing. So what you will end up with is those vesicles. So this is a TIL series that we can record from our sample, and this basically we record a tomogram. So you tilt your object and record a short exposure. You have to keep your dose constant because otherwise you'll burn your sample. And then you don't see anything in your TIL series. You only see that you have some membranes and those little gold beads that you use for fiducial alignment. Then as a result of computational processing of those TIL series, you get a tomogram. A tomogram is a 3D volume where you've back projected all your TIL series to a 3D volume, and then this is a 2-dimensional slice, thin slice, through a tomogram, right? So it's a 3D volume, but this is just a small part of it. And in this tomogram, you actually can see molecules. Well, you also can see membranes, but these large molecules over here, if you look at them from the side, which you can do because your data is 3-dimensional, they would actually represent ryanodine receptors. They would be large, of the proper size that ryanodine receptors are, and they would also be tetrameric. So you don't see a lot of information in those because you're limited by noise that you by the amount of electrons that you can apply to your sample, which results in noise that you still have present in your tomograms. But what you can do, you can extract those molecules in boxes, mutually align them and average, kind of similar to single particle Cryo-EM, only the whole process happens in 3 dimensions, and that's why it's called subtomogram averaging. So you have a tomogram, and then subtomograms contain your molecule of interest, and then subtomogram averaging is that when you produce an average of subtomograms. so we tried to get the structure of the ryanodine receptor next to this membrane, which we thought that sometimes would look like T-tubules. However, most of the receptors look a little bit like this, and they're not attached to anything. And it's probably because during the purification, a lot of the interactions have been broken. We're still trying to get this more particles to average, but so far we don't see in the average of the receptor with the membrane, we don't see the DHP receptors. But we could get first the structure of ryanodine receptor at 13 angstrom resolution. That was nice because we could visualize the membrane, and we could see that the extracellular domain, I showed over here, actually fits. So the structure fits rather well to the structure that have been purified and solved by the Joachim Frank group and the other groups as well. Interestingly, what we could see is that the membrane was not horizontal, which everyone would draw it to be, but it's a little bit bent. And the curvature of the membrane, you can see, is higher. So the membrane in the average is higher, is curved stronger than of the vesicle in which the protein is present. And even if you can see that, if you can look at the individual receptors, you could see that the membrane would not be smooth around them, but would be a little bit unsmooth and tilted. So that's curious. We don't know why that happens. We would need to have a high resolution structure for that. But what we did, we added a ligand, ryanodine, which would lock the receptor in the open state. And then now we see the toggle between the structure in the open and in the closed state. The open state is a little bit lower resolution. So what we see that the outer domains move similar to what was shown for the single particle cryo-EM structures, but it also seems to bend the membrane. And so with the open state has the membrane even slightly further bent. So yeah, obviously we would need a high resolution structure to understand why that happens, but the effect that it probably has is that there is an elastic energy stored in the bent membrane, and when the receptor is open, this membrane wants to bend back and would have an effect, act in the direction of closing the receptor, which was not shown for the purified receptor because it didn't have the membrane. Okay, so then we decided to really go and try to get high resolution of this receptor, because the first structure was, well, we just got all we could do, but then obviously with tomography you have an opportunity to significantly improve on your data collection, data processing, and throughput. So the first thing we did, we decided to do the so-called hybrid subtomogram average in Cryo-EM. So what we do, we first record a high-dose image of our object of interest, and then we record a tomogram from the same position. We merge these data sets, and then we first do subtomogram averaging as usual, so that gave 13 angstrom, and then we do the reconstruction only from the high-dose image. And then we can also refine the high-dose image against the structure, which gives us a little bit of improvement in the resolution. So that got us to 9 angstrom, and at 9 angstrom we can see alpha helices, which basically the protein is mostly alpha helical, and we can see most of the helices. And we could see that some of the helices actually don't fit the density very well. We were very happy about it, and we first tried to do the structure of essentially the receptor in the open state, but we didn't get to that high resolution yet. Good thing about this paper that got us really excited is that essentially we thought that we're limited now by number of particles. What we do here, we do a single particle reconstruction from very thick ice from very small number of particles. So it's 2,500 particles times C4 symmetry, it's 10,000 particles. So essentially if you do a single particle reconstruction from 10,000 particles, you're not going to get a very high resolution. So essentially what we thought we need to do is we need to get more particles, and then we probably can get to higher resolution. So then we had to, it's not that easy, because subtomogram averaging is a big process, right? It contains a lot of steps. Some of them are rather manual, and at this EMBO workshop where we come from, we actually had to go through the steps of subtomogram averaging, and for some of the students they were overwhelming. And you ask Pushkar later how that is. Good news is a lot of these steps can be done automatically, but they usually are done automatically by a dedicated software piece. So you need to have like those scripts that then would go from one step to another, and then this is just technically difficult, and you have all those scripts that are ugly, and then some student leaves the lab, and then you cannot reproduce them anymore. It's a difficult problem, like everybody knows this, right? You know, you sacrifice the speed of execution by speed of development, and then and then this house doesn't hold very well. So yeah, my student Nikita, he was a computer scientist in my group in Frankfurt. He decided to invest his time into automation of the workflow, and he designed this basics for electron tomography and automated reconstruction, which is a TomoBear, to interface common EM software and to design it with an ambition to run it in an automated way. And it kind of worked to a certain extent. What it did is basically allows a lot of outsourcing of a lot of human intervention, starting from the data that comes out of the microscope. This is roughly how it works. So tomograms can be produced almost automatically these days with this gold bead tracking. And so after that, we need to identify our particles of interest in those tomograms. You probably don't see where those particles are. I see because I worked at this for a long time. So, but this is roughly how data looks, right? These are the tomograms. So if our protein is large enough and we have a template, then we can do this cross-correlation search. So we can rotate our template against in every possible rotation, and then kind of score how well it fits to every position on a map, and then you will have those high cross-correlation parts, and then you just kind of can threshold it, and then that would be the peaks that we would want to extract. So this would then would correspond to the particles. So we essentially can pick particles for the ryanodine receptor almost automatically because they are very large. And then what TomoBear also allows us with this reasonably simple command to do the first round of subtomogram averaging classification, right? Because obviously with this procedure we're also going to pick some rubbish. So we will set noise traps, and then we will run one round of alignment to have good particles and bad particles separately. Then we can take the good particles, throw away the bad particles, and continue to refinement. With that step, automation essentially is over, and the user has to take over. But this saves a lot of time. And I started my lab in Frankfurt a year and a half ago, and I was really happy to see how, after a couple of months, people would get tomograms and some structures, and I was really excited about this. So that it works. So we benchmarked it on some data that we had. So we recorded the apoferritin tomogram, which is a CryoEM crime, and which is a tongue breaker, CryoEM crime, and we could, with almost automated processing, with essentially only the class selection for 3D classification, we could get to the Nyquist over there, which was 2.8 angstrom. And then we also reprocessed our ryanodine receptor dataset from the 2020 paper. Got to slightly higher resolution, but I don't think this is very important. Essentially, the important thing was that both structures were done in a week time. So the automated processing lasted for a couple of days, and then the user took over, and we run it in Relion 4, which is a beautiful software for the last to squeeze the last high resolution details out of it. And that actually works very well. So now we think we have a good toolbox. The only thing that we have a problem with Relion is it's very slow. When you get to a very high resolution of a very large molecule, then your pixel becomes very small, and you have to and you end up with a very large box. So when you calculate cross-correlation between your template and your particle, you have to rotate and Fourier transform them many times. Basically, doing the angular search, so you screen the angles, and then all you do, you rotate your template and you do Fourier transforms. The larger the box, the most computationally expensive it becomes. So I want to also show you a little software package that my student Ricardo released. So essentially what he did, he replaced the order of operation in calculation of cross-correlation in 3D. So what we typically do in tomogram averaging, we have a tilt series, our projections. Then we reconstruct the tomogram. It's a large 3D volume. From there we crop particles, which are also very large, and then we rotate the template to do 3D cross-correlation. And then we do this for all the angles. And then we calculate cross-correlation. So what Ricardo suggested to do, so we know where each particle is in every projection and at which angle roughly it is there. So he extracts a sub-stack for each particle, and instead of a 3D cross-correlation, he calculates a set of 2D cross-correlation with a reprojection of the template. Essentially, here he tweaks the computational complexity and only does the 3D reconstruction of the cross-correlation volume in the area where we need it. So essentially, the computational complexity goes from the purple line with the conventional cross-correlation to basically roughly very small values, and this is the same curve in the logarithmic coordinates. It's essentially what we could never do with the conventional cross-correlation algorithms, we can now do with Susan. And obviously it also gets high resolution. So in this work we use Reliant 4 because we wanted to have it separated. And then Vasily was really happy was really enthusiastic to use Reliant 4 because it just came out there, right? And then by using a larger data set that we also collected in collaboration with Thermo Fisher, because they offered us an opportunity to use their super best, super new hardware, which actually made a bit of a difference probably. We got first to 4.6 angstrom for the entire map, and then Vasily did the local refinement of the transmembrane domain and the core domain of this protein up to a resolution of 3.9 angstrom. At this resolution, you can actually see very nice side chains, and then you can refit the available atomic models into those densities, and they fit rather well. So these are the most important transmembrane helices. We were very happy about it, and I was a little scared, like, is it actually different to the structures of purified receptors? Because, as you've realized, we've put all this effort over many years. The way to look at this, in order to look at that, please consider the activation mechanism that was suggested by the Cell paper in 2016. So there is an apostate without ligands, and then one of the ligands, calcium, is added, which sets the receptor to the so-called prime state. Here the pore is still closed, but it's more ready to open. And then when another ligand, the physiological ligand is ATP, is added, then the receptor opens the ion pore. And then it goes through an inactivation state to basically, it goes through an inactivation state. So what we've done, we've taken all these published atomic models, and we calculated distances between each other, and then we projected them to a 2D plane, which looks like this. We color mapped them, so open states that have been reported are in green, apostates are in dark blue, cyan are primed. And then we can see, basically we can just overall see that there is certain consistency here. So first priming goes from dark blue to bright blue, then activation goes from cyan to green, and then inactivation goes to the yellow structures over here. And the yellow structures are slightly different than the apostate. So we only have an apostate so far, and it's one structure without classification, and it comes out here. So this means that basically our apostate in the native membrane looks a little bit more like the inactivated state. And the main differences are in this domain over here that touches the membrane. I don't want to go into too much of the biology here because we're still working on this, and I think it's also important to not have just one structure, but to have several structures. We're doing classification and trying to get the classes to high enough resolution that we could build in them, see how far Vasily can get. But the good news is that the structure is actually significantly different to the structure of the purified receptors.
Yeah. This alpha RMSD is for the overall structure or just for some ratio?
So this, the way we produce this is we align all the structures by transmembrane domain, and then we calculate the RMSD only for this core domain, excluding the outer regulatory domains, because they are the largest. Yeah.
So you have the best resolution in that region.
Yeah, and we also have, obviously we're more. We have best models there.
Right. Got it.
But it's C alpha RMSDs. We don't look at side chains.
Right.
But the scatters look very consistent, and we can do this for all the domains. But we think that this core calculation for the core domain, core domain plus the transmembrane is just more relevant because it's how the activation happens. So I guess we still have some work to do here in order to basically understand if it's just if all the structures are here or they're a little bit spread. And so we're working on this. But the good news is that yeah, we can solve the high resolution structures. And so what I wanted to emphasize is that it's not a native tissue, but this is a native membrane, right? This means that it has a native lipid composition. It has ryanodine receptors have neighbors. So I didn't show it, but obviously they are very, very densely packed. And we're very happy that the structures are a little bit different to the ones that were purified in the detergent. Now, in order to expand our approach of imaging proteins in native membranes, we came up with this little trick. So we express, and this is for the serotonin receptor ion channel, which I showed you in the beginning. We express those proteins in membrane vesicles, well, in cell culture, and then in the cell culture, cells produce membrane vesicles, which we can collect and put them on the grids. So we don't do any protein purification, we just only purify the vesicles from the cell culture supernatant. And then we can label our protein of interest with gold beads, and we can see that some vesicles have a lot of labeling, some vesicles don't have a lot of labeling. So we can record data on only those labeled vesicles, and then we can see those little barrels on the extracellular side. I marked them with red dots. So these look very much like the serotonin receptor, and then they also sometimes have those gold beads that I computationally erased from these tomograms because otherwise it gets too crowded. But yeah, essentially the labeling is quite specific, so we think that this is the right protein. And then there are a lot of advantages to such approach. The only disadvantage is the obtaining meaningful resolution is difficult. So Xiaofeng spent a year and he got this. It's roughly 10 to 11 angstrom structure. It has all the right features of the receptor. We so far cannot resolve the transmembrane helices, but overall this is the correct structure. So I think, you know, we first, for the ryanodine receptor, we first got to 13, and then we got to 9, and then we got to 4. So I hope that, you know, with a slightly improved data collection scheme, we can get to slightly better resolution, which we're working on now. And then we obviously will be able to say what is the real conformation, right? And then how it really, we can probably also work a little bit on the real opening. And here we will not have an impact of the nanodisk in here. So we would also obviously can say a few words about which nanodisk is the best, which will probably be a little bit specific to this protein. But yeah, overall I want to encourage everybody to try to express your membrane proteins in such a system and learn tomography because this way you can get the structure of your proteins without purification. You save on purification, you get more native structures. However, it will probably take a little bit more time. Right? So that's my last slide. We can see strong effects, not only us, but the entire community of membrane protein structural biologists. We can see effects, and in this case, we also see some non-trivial effects like bending the membrane upon activation by ryanodine receptor, and we can solve structures in situ, and this way we can actually address some of this regulation by lipids. Yeah, and then obviously these structures are more physiological. It's a little bit difficult, still requires optimization, and so we're working on it to make it easier for ourselves, but also for everybody else. And then in terms of technology, we're now looking into the application of machine learning to make this whole process even faster and a little bit more reliable. So I want to thank the people who did the work. So Yini and Patricia did the work on the serotonin receptor together with our colleagues from Shenzhen Institute of the Chinese Academy of Sciences, is Shuguan Yuan and Horst Vogel, that are our colleagues. And then Vasily and Venbo did the work on ryanodine receptor together with Nikita and Ricardo, who did the software development. Thank you for your attention. I guess students first.
Yeah. I have one question. When you showed the last image of the vesicles with the receptors out, how did you manage to get the gold particles exactly? I mean,
well, it's a labeling with
so the gold nanoparticles.
Yeah. Well,
how did you manage to deposit them exactly on the?
Well, these are functionalized gold beads that bind to the strap tag, which is on the protein. So the protein has a strap tag. You actually see it over here on average is diffused, and then gold beads just bind to it. And then some vesicles have a lot of receptors, some vesicles don't have any receptors, so obviously it binds there. It's just to show that you can if you work a little bit on your labeling, then you can actually say that this is my protein of interest. You think that this is a lot, but actually only maybe 5 to 10% of receptors are labeled, which is nice because you don't want them all to be labeled. It's too much gold.
What are the number of tomograms that were used for this and the number
Well,
of particles for averaging?
I think it's like several hundred tomograms with, I think in the end, Xiaofeng ended up with slightly less than 10,000 particles with C5 symmetry. So yeah, we can improve on it, but for example, you can see that you still have like an edge of the carbon grid. So if we now switch to gold grids, that should give you a little bit less motion. So we also basically improved our data collection strategy a little bit. So I think that data will be better, and we can collect even more tomograms now.
What till time we have able to collect the data in this case?
So when you collect this so-called dose symmetric tomograms, then you essentially can collect to plus minus 60, and then you can throw away the bad tilt series, the bad projections. So we probably go to 50, plus minus 50.
And burning also happened in that case because
Yeah.
production of tomograms will take a little longer time, more than even thrice or 4 times.
Yeah. So again, tomography has the same thing as you know. You always need to compromise maybe. So if you want to apply higher dose, then you know that you will have more sample damage, and also all your sample is going to move a little bit more. So it's an experimental parameter that you have to figure out a little bit for yourself. But yeah, so with a dose symmetric tilt scheme, you spend more time recording the data, but it saves you the effort of thinking in retrospect that maybe you could have applied more dose.
And also depending on the
Okay, while doing this, the major problem has been that whether its structure is real or if there is some artifact, something is coming or not. But if you, let's say you add some ions, which are the ion channels, so there are ions, or you add the substrate or some of the inhibitors, and then do together some of these things, can't you get a real picture of that?
Well, it's a great question. So I'm not saying that this other structures are artificial. I just don't know which ones of them is correct.
Correct.
So yeah, if you add ions, so obviously, for example, in the potassium channels, you see the ions very well positioned, in the selectivity pore. In these channels, they don't have like this defined steps, so they just go. You've seen the AMD simulation; it just goes through it. So they will not be ordered, and as a result, you are not going to see the density of them. So this probably will not be very, very good structure.
So, I think she has a question. You go ahead, but please go ahead. I can
Question which was asked by sir over the nanodisk. So if your membrane protein is bound to the nanodisk, do you think that it will lead to a biasness in structure reconstruction, like it is in a particular conformation which will be preferred, which will be more in the connected phase? So when we are building the structure, it will lead to a biasness in the structure.
Yeah. This is what I think this paper showed, and it also depends on the type of a nanodisk, right? Because I would hypothesize that, for example, in this nanodisk, the interface between a nanodisk and the protein transmembrane helices is affected by the type of the nanodisk, right? So, but how exactly that happens, it's hard to figure out, and then it's probably specific it's probably protein specific too. So, but yeah, as with Rebecca's talk, you need to take your structures in nanodisks or any structure a little bit critically and see what could potentially affect the conformation, right? But yeah, excellent thinking.
So I asked the question about the serotonin receptor because the field thinks actually that there is an unblooming movement in the transmembrane domain. Basically, you know, there is a vertical and a horizontal translation, both happening simultaneously. So probably a vertical movement and then a horizontal movement in the anti-clock direction, which is leading to the opening of the channel. Now this observation of yours, which is actually just horizontal widening of the TMD, if that is true, I mean, like, it's going to actually have a large impact on the whole landscape ion channel community. Because it's a revolutionary thinking, basically. Like it will change the thinking.
I mean, I've seen this. I agree. I've seen the. Yeah. Okay. You see, this basically. I've seen something similar.
This is called unblooming.
Yeah. So I've seen a similar conformational changes in some of the structures in presence of nanodiscs. So I'm not saying that it's just a nanodisc that just solves this problem. It's probably more in the biochemical protocols that in a protein-specific manner can influence it.
Yeah, I think the CryoEM would definitely answer this question, because I mean, like when you're doing it in the native membrane preparation, that's when we'll probably get the best answer. And one more thing, I mean, like, do you think since the number of particles is actually a bottleneck by which you could reduce the, get the better the resolution by enriching the membrane with the proteins by whatever method, will that help?
So in our approach, we just overexpress the protein, right? And whatever goes into vesicles just goes into vesicles. So you don't, so you can enrich the vesicles, but I think it's very hard to enrich the protein amount. Or?
This is the first, this is the way when first time actually the structure was solved for ECHR back in.
Oh yeah, Nigel Unwin.
So
Yeah.
he used an enriched expressing membrane, right? So of the string rate, so electric rate. So do you think, I mean, that kind of approach would be really helpful?
Well, I think Nigel Unwin brought it to an extreme because they formed crystals.
Yeah.
I would kind of try to have it non-crystalline. But it would, so what we're trying to do is we're trying to basically increase the expression levels rather than play with the concentration. I mean, once you have those vesicles, you already cannot do much with them. You have the amount of protein that you have in them. But yeah, so the enrichment step would have to be before.
Yeah, before.
And it's a cell culture expression system, so I'm not sure what I could do there. I think what we want to do, we want to compensate with throughput, because we can do it these days with good microscopes.
Very good talk, Mr. I have a small question related to the technical one. Suppose that there is vesicles for labeling you are talking. This kind of work can be extended to understand some organelle bound protein, just to isolate cell organelles, just like a mitochondria, and see that any effect on the drug.
Yeah, I mean, it's basically, you can do, there is no rocket science there. It's just you get fractions of it. If you can purify mitochondrial fraction, you can also do the same thing. It actually has been done. If you can have an extracellular tag, you can bind whatever you want. If you can genetically modify your protein, you can just express an extracellular tag.
So we just suggest that if you go directly, or we go with the lamellar preparation and then do this.
So lamella is great for a lot of reasons, because it really gives you all the context of the cell. But it's more difficult to image, it's lower throughput, and typically it's harder to get high resolution. Also, you get less molecules per field of view, right? Because you have thin ice, very thin ice, and on the top and on the bottom you probably have them partially damaged. So we can discuss this later, but there are pros and cons of both approaches here.
Are there any more questions? Okay, there's one last question.
With the nanodisc receptor, where you see that the bend in the membrane. So you seem to suggest that the membrane, that the change in the membrane bend is due to some stored elasticness. But I'd assume the membrane should be a fluid thing. And how can it store elasticness?
Well, I mean, it does bent membrane do tend to unbend back. Like, I don't know, like my membrane biology that I remember suggests that they don't like to be bent for a very long time. So we haven't done the calculations. We actually should team up with a good theoretical biology group and do the calculation what would be the force that like to which extent the membrane could bend, how much, and what's the energetics of this. We haven't done it yet. It's just my intuition, and it's a little bit of a speculation, I must say. Thank you.
Thank you very much, Varsha. We will go for a quick trip to our new Cryo-eM facility. I'll request all the students to wait outside LHC, and we will take you in small groups because everybody can't fit in there at once. But we will all go, right?
Great, thank you. Thank you very much. It was excellent discussion.